Welcome back. Today we are going to look at complement fixation. Complement fixation uses classical activation of complement system as the principle on which it is based. We know that classical activation of complement system uses antibody-antigen complex to initiate the activation process. So we'll have that as an antibody and that as an antigen. So this is antibody, this is antigen. So they should be specific to one another. So let's call this antigen X, and antibody to the same antigen, call it antibody X. So this one is now an antigen antibody complex. Antibody antigen complex. So complement fixation is used to find out if a particular antibody is in the serum of a patient or a particular antigen is in the serum of a patient. So as we stated earlier, to do this, you use either commercial antigen to probe or test for an antibody in the serum of a patient, or vice versa, you can use commercial antibody to find out if its specific antigen is present in the serum or body of a patient. A good example is, let's take Salmonella typhi, which causes typhoid. A patient is infected by the bacteria, and or you suspect is or she is infected by the bacteria. So you know when the bacteria get into the body, it causes production of specific antibody to itself. You go and buy commercial antigen to Salmonella typhi. You have it ready. Then when you come, you take venous blood of the patient and obtain serum from it. The serum should contain the antibody to Salmonella typhi. If you mix the two, since the antibody in the serum is for Salmonella typhi, they are specific, so they will form that complex. That's what we are talking about. But in complement fixation test, you will have a patient serum. You will have a patient serum, two. So there you expect the antibody. Then two, you'll have commercial antigen, then three, indicator. Substance. or reagent, electrical substance or reagent. So you'll have those three. So to begin, we'll start by a case where we assume the patient's serum as an antibody. So I'll draw a test tube. So this is the serum. So it has antibody let's just use X. So your commercial antigen should also be antigen X. Since they are specific, they will form antigen antibody complex. So this one is the initiator of classical complement activation. Then I add a complement. Then I add a complement. Then I add a complement. So when I add the complement, when the complement reaches this mixture, it will get the complement will get this complex, antibody antigen complex. So there will be classical activation of the complement, and the complement would be consumed, or what we say, fixed. It is fixed, it is not there. Once it is activated, it will no longer be there. We remember what happens due to complement activation. So that's the first case scenario. So in that case, let's do another tube here. So this tube has no complement. It has no complement because the complement has been used up. So this tube now has no complement. We can start from up again. I have a tube in which I have a serum which contains antibody X. Then I add antigen X. They are specific, they form antigen antibody complex. Then I add a complement solution. It will be fixed, it will be activated by classical activation, and then it is used up, it will not be there. I have that first case scenario. The second case scenario is where I add another antigen. I add, instead of this antigen, instead of antigen X, I add, I didn't put it here, I add antigen Y. Instead of X, I add antigen Y. So the first complex was between antibody X and antigen X. Now I have antibody antibody X and antigen antigen Y. So in this second case scenario, there is no antigen antibody complex because they are not specific to one another. This is X, this is Y. So there's no complex. There's no complex inside the tube. So there's no complex in that case scenario because we have an antibody, specific to antigen. So when I add the complement, to that tube now, it is not consumed because this thing is not there. Instead, we have separate antibody, separate antigen. There's no complex. So there's no classical activation and the complement remains. So that's this case scenario now. I have a tube there. This tube now has complement. That's complement. Why? Because there was no activation, not used. So I have two tubes. So what will follow is that we'll add now indicator substance. So the indicator substance is another antigen antibody complex, but it is different from what we had earlier. It is already prepared. So normally we take sheep red blood cell, and produce antibodies against sheep red blood cell. So those antibodies are used to sensitize the red blood cells of the sheep to form antigen antibody complex. So I'll draw the indicator substance there, just sheep red blood cell with antigens on the surface. So each of these antigens are complexed with the specific antibodies. So this is red blood cell, which already have attached. So just like this one, Mm. Just like this complex, this is antibody, antigen. This one is antigen, this whole cell. Then there are antibodies attached to it. So it is antigen antibody complex. So if it is put in a system where there is complement system, it will cause activation. So you will add this solution or suspension of this complex into this and that. You'll add it to this test tube which has no complement and this one which has a complement. So when you add it here, there is no reaction. So there is no complement. The red blood cells remain intact and you can view them under microscope. When you add here, there is complement inside here. There is going to be classical activation of complement system. When that is done, that is occurring on the surface of this cell. And we remember what we call the MAC, membrane attack complex. It attacks the membrane of the red blood cell and they burst open, what is called lysis. So this one forms a red solution, while this one you see the red blood cells. So that is the principle of complement fixation test. So a positive test is where you, at the end, you see red blood cells. That's a positive test. A negative test is where there's lysis of red blood cells. So we'll explain more about this, but today we are just talking about the principle. At the end of the principle, remember positive test, there's no lysis at the end of the tube, while negative test, there's lysis. Thank you. Let's meet next time.